joke about Acme Corporation, but I don't know how many people watched Looney Tunes that know that whole thing. Just me. He took a little detour to Ann Arbor and then back out to San Diego. I think, I didn't ask him, but I suspect he developed a passion for neuroscience when he was in Michigan and changed track a little bit. And then after his PhD, he did 2 postdocs in Boston, first at Tufts and then at BU, before he went to Cornell as a research associate, working his way all the way up to full professor. What's that?
Ever so gradually.
Ever so gradually. It looks pretty timely to me, in the Department of Psychology at Cornell. I knew that Tom had spoken in 2013. Amber had sent me a list of the speakers as far as she had it. I went through his CV and I saw he actually gave a seminar in 2003. So this is his 3rd time, and he comes every 10 years, and you can start making plans for 2032 or 2033. But if you look at the titles of the 3 talks, it's very interesting how he's gone from, very reductionist look at olfaction and has worked his way into some incredible complexity. And that's what we're going to hear about today. So, Tom Cleland.
Thanks so much, Bruce. Still feels a little transgressive not to be wearing a mask, so I'm sure I'll get used to it fairly quickly. Yeah, so thank you, first of all, for coming, for making the time. And I guess I'm going to try and, if there's a fair amount of detail, but I'm going to try and focus on these sort of 3 points, which is really what I want to talk about today. The first is to sort of convince you that the regulation of spike timing at this milliseconds timescale, on the gamma beta oscillations timescale, matters, happens and matters. I'm going to make try to also convince you, more just by assertion, that we're all interested in sort of how these sensory systems encode the outside world. What I'm going to sort of assert is that that simple replicability in sort of clean environments, about getting the same odor twice and getting common input, isn't enough, because these systems operate in the real world, where the primary receptor systems that the patterns of which are on what odor quality depends are really irreducibly messed up by background odors competing for those same receptors as antagonists, agonists, and such. And so if we want to think about a good system for odor representation, we need to take that into account. How does that get fixed? And what I'll then tell you about afterwards is a sort of theoretical model that sort of puts it all together about how we can use this principle to solve that problem. And it's both sort of a computational neuroscience model that I would like to market as a way to think about how the biological system works, but a substantial amount of it has actually been instantiated for artificial systems on neuromorphic hardware as a way to sort of have artificial noses that can cut through unpredictable sources of noise. Okay, so the first thing I'll talk about is actually not data from my lab. I don't know if Kevin Boulding is listening right now, but these are actually data that he generated, and he and his then PI Kevin Franks posted this as a public data set on the CRCNS website. And my student Michael Mariscal here has been analyzing it for these fine timescale dynamics findings. So these are just 2 examples. There's a fair amount of heterogeneity, but some core principles arise. This is where these are all, it's a very rich data set. I'm not going to be showing you most of what is possible to glean from it, but these are odors that are delivered at this time. This is something what we're calling early beta, which sometimes happens. It's, I don't want to get too excited about it. It's new in a sense, but I don't want to get too excited about it. I suspect it's just a reverberation through the dynamical system that happens because of the sudden encounter with the odor. Some of us will be familiar with gamma oscillations, which are endogenous to olfactory bulb, and then beta oscillations that follow them, which are particularly coherent back and forth with the peripheral cortex. And I think of that synchronization as being the mechanism that enables their intercommunication, their meaningful intercommunication. Sometimes you actually don't see the gamma arise. I just wanted to put that up there as an honesty check. A lot of this is probably because of the details of electrode placement. Of course, we have hundreds of electrodes to work with here, and this is just from one. But so not all electrodes will show everything that is going on, but it is nevertheless a variant we see. But these beta oscillations between the piriform cortex and olfactory bulb are prominent. So this is a map of sort of average across all animals, odors, and trials, which brings, which smears over some of the subtleties. But I just wanted to point out that I haven't been cherry picking examples. We have this very powerful quote unquote early beta, then gamma oscillations up at this higher frequency, then followed by this extended beta period, which is more powerful than it looks. This is a very different scale here than this scale. But again, the principle here is just that that sequence of sort of oscillatory domains is common and replicable, a bit heterogeneous, but basically stereotyped. And furthermore, between olfactory bulb and piriform cortex, most importantly, the beta, but actually really all of those signals are coherent. So in other words, oscillations that are happening in the olfactory bulb are coherent with those that are happening in piriform cortex. Not exactly at zero phase, but tightly coupled with one another. But more importantly, what I'm interested in here is not the oscillations per se. Those are kind of side effects of coordinated dynamical systems, but rather about the actual spike-spike interactions. This is up here is simply showing that the action potentials of mitral cells are constrained with respect to phase, but the most important thing I want to actually talk about here is not that this is universal, weakly universal. It is extremely strong and partial. There are a subset on the order of 10%, 15% of the mitral cells for any given odor, and that'll come up in the slice work later, that are strongly coupled in an odor-specific way to this underlying oscillation, and a larger number that don't care much and do not align to the oscillations. So what you see here, this is essentially a statistical diagram where the bias of these these oblong circles from a perfect circle is a measure of their bias towards spiking at particular phases of early beta, gamma, or late beta oscillation. Down here, somewhat more complexly, but more interestingly, is where Michael developed this system or implemented this system where for every spike in a neuron, every spike in a mitral cell, you say, around this spike, what is the mean rate of all other cells, all other mitral cells? But you filter this through the subtraction of two kernels. So it's this measured with a 3 millisecond kernel, very narrow in time, minus a broader 11 millisecond kernel. Lots of details, but the outcome of which is you're basically asking for every spike, how likely is it to be tightly synchronized in time on a millisecond time scale, 1 or 2 milliseconds apart, with other cells, and how much is this bias with respect to gamma phase? The fact that these are strongly pulled over here tells us that there is a process that is aligning many spikes to one another, specifically to a given phase of gamma. Sort of just directly showing what is sort of implied by the very existence of gamma, that things are happening in coordinated fashion that shape spike times and as a byproduct cause these field potential oscillations. What we're seeing is action potentials that are more aligned to one another, more coherent, close to one another in time, more synchronized than would be expected by chance. So first of all, we wanted to take this down a level. So I'm going to show you a bit of slice work where we're now talking about the olfactory bulb in the slice preparation. So we're only going to see gamma, not the sort of beta interactions in the piriform cortex. But our goal here is to look down for some mechanisms about what, how do you generate these oscillations? What do they mean? How do spikes get aligned to them and such? And then I'll talk about what we can do with that. So this is Jesse Wirth and Matt Einhorn. Matt is sort of our engineer who masterminded the reconstruction of this rig here. What we're doing here is creating a slice preparation where we can deliver artificial odors. What this means is that we have a sort of out of focus slice here in which OMP containing neurons are expressing channel rhodopsin as well as yellow fluorescent protein, which means that we can shine light on them and stimulate them as if they were sensory, stimulate the axonal arbors of the sensory neurons that persist in the glomeruli. The software that Macro lets us sort of build these spatio-temporal profiles of stimulation that can act to whatever extent we want to program as artificial odors with appropriate time scales and appropriate complexity depending on the question. And so the data we get out of these are not optical data, they're electrophysiological data from a microelectrode array, which is right here, in which you see, in this case, you're seeing the external plexiform layer going along here, which has a lot of odor-gated activity on it, both spikes and field potentials. Here you can see the spikes. These are just 3 candidate cells to show you kind of the diversity of cellular responses to these artificial odors, looking sort of a great deal like the variety you see in the sort of classical papers about electrophysiological proteins and microcytes, where the odors onset and get some excitatory responses. We don't get very many inhibitory responses, but there are some because the getting spike rate is so low in part. And this is probably not the case of that, but we do see some probable inhibitory responses. This is sort of what it looks like, what one of our stimuli looks like. This is sort of a 2-spot stimulus. It actually goes to about 6 hertz. And this is slowed down for So as every basically what we're doing is thinking about these slices. We're looking at sort of artificial odors in the sense of what this circuit of the slice might see from an odor with the appropriate timescale dynamics, which are important. And the first things I'll talk about is just showing you that this works. So here we have I'll talk a lot about sort of odor A and odor B. Again, these are actually patterns of light. These 2 spatial coordinate sets here at a low concentration, quote unquote, less light, and then at a higher intensity here. And what you're sort of seeing is that particular, I'm just going to keep calling them odors, so forgive me, these are patterns of light. You see that then when we stimulate, we get these responses and this sort of responsive neuron 1, but the same neuron does not respond, interestingly, to odor B. Whereas neuron 2, just another one recorded in the same slice, the electrode array, doesn't respond much to A. It's actually, we suspect that's an inhibitory response, but again, that's really statistically difficult to ascertain, and a mighty response to odor B. So we're creating these artificial odors are differentially activating mitochondrial cells. Yeah.
I sort of lost the plot here. So how do you get this selective expression in the
we don't get selective expression, we get selective targeting. So we only activate these different spots with our
Oh, okay.
That's the killer aspect of this is being able to say, okay, we're going to build this arbitrary set of shapes and with the different spatiotemporal profiles and say that's odor X. Okay. Thanks for any other clarifying points I could make. This is just a walk again, sort of a testament building slide to show you that this is a spectrogram, which is sort of showing the frequencies that occur in time. If things are just arbitrarily noisy, you won't see solid patterns here. But this tells us is that the delivery of this odor generates a predominant frequency at about, in this case, 40 to 50 hertz. Right, this is like invisible. We're still talking about local field potentials at this point that carry on for a certain time, and you can see that they create this peak in this power spectrum here, showing that this frequency sort of rises above the background noise that the mixture of frequencies expressed. But again, it's more important to think about what the individuals, is this indicative of spike timing regulation? And here's an example of what I was talking about earlier, where if we look at all of this, the cells being recorded from one given slice, this gray phase plot here is saying, what is the distribution of their spikes with respect to the phase of gamma? In other words, if this is the trough, then this is halfway up, this is the peak, this is the trough again, this is again or sorry, this is halfway down, this is the trough again. Where are those spikes? You'd expect it to be pretty round if there were no effect at all. In fact, this is significantly different from round. But it also shows you that most cells aren't paying attention. Only a subset of neurons are actually aligning. And if we cherry pick a couple where we show some good examples, they look like this green one here, which once the stimulus comes on, snaps to and fires only in strict phase with gamma. Some cells do that in an odor-specific way, but not most. Here's an example of a cell that does. We're now looking at its degree of phase locking as a statistical measure. So it's statistically significant above the green line, statistically very significant above the green line. And so this plot goes along, and when the odor comes on, you see this rise up to being tightly phase constrained with respect to gamma. The other odor B, doesn't. The lower concentration, lower intensity of A does, and B does not. And notice in these cases, the spike rate, the instantaneous spike rate is not changing. This is not just I'm increasing my activity. This is, in this particular cell's case, not really changing its mean rate, but just locking to gamma phase. This is again illustrating that the baseline, despite the fact that stimulus is going on, many cells don't really care about it, but a fraction of cells care a lot. And again, with the same sort of statistical measure, this is P, anything above this is P less than 0.01, is strictly phase aligned to gamma. And this just shows, if you see the rhythmicity down here, that when the stimulus is on, you have this rhythmicity is spiking following the rhythmicity of gamma. So the last slice piece is really just to say, let's get, we talk about gamma because it's the evidence that it was the original evidence that there was such a clock, that there was such timing regulation. But it's not something that gets communicated to the next level. Spike times will go to the next level. So let's talk about this in the sort of the kind of the absence of gamma. So what we have here are just two arbitrarily chosen cells that both happen to respond to the same artificial odorant. And so they're different, their spike trains are different colors here, which is a little bit hard to see. What's easy to see are these black rectangles. And that is the times at which two cells are spiking in strict alignment, where that means plus or minus about a millisecond. And in this case, the quote-unquote artificial odor is delivered, and you get somewhat of an increase in spike rate, but primarily an increase in the synchronization of the spikes between these two cells. And this is sort of quantified down here as there was a little bit of an increase in spike rate, which is this black line here, or the spike rates literally are here, but the black line here is what coincidence rate in this fine window should we expect given that higher rate? And if you are significantly higher than that, then there's an additional process that is constraining your spike times to be more similar than they would be by random limit. And so we see actually a more impressive than it looks factor here, and a truly ginormous effect here. The blue being over the black means that much more than would be predicted by spike rate alone, this pair of cells is snapping their spike times aligned to each other with respect to the phase of gamma. So not just the respiratory phase, not even just in the same gamma cycle as fasted net, but phase with respect to that gamma cycle. Again, the time cycle of a small number of milliseconds. And this is up here showing us, if we take all, again, these are two individual examples, if we take synchrony matrix, so that for all possible pairs of units in a given slice, what is the matrix of their unexpected coincidence rate for both odors A and B, and for their two intensities, low and high, and reduce that into a principal components matrix here. And you'll see that basically both the blue, the high and the low concentration of B, and the high and the low concentration of A cluster together. What this means is that despite the fact you can, there are many, well, many being like, I think 6 offhand, mechanisms from the periphery to the bulb that are essential for the broader problem of correcting for concentration, for segregating quality from concentration. It has to happen at the early levels or it's not going to work. But one of those levels is clearly seen between bulb and piriform cortex, where you can still see concentration effects in metral cell responses to a much greater extent than the piriform. What this is telling us is that once you map this onto a synchrony metric, it's done. In other words, piriform cortex is, if it reads the synchrony metric, which it does, we know that from Luna and Chapa, then that's that we're essentially rid here of the last vestige of concentration information. That's too strong to say. There's almost something that leads to it. But at this point, different intensities are just as replicable as repeated identical stimuli coming out of the bulb. Whereas if you actually change the quality from A to B, you get a greater difference between locations. In other words, concentration and quality have been effectively separated, at least within the limits of the diversity of stimuli that we've used here. Okay, with me so far? So this is showing that these are, that you get particular samples of mitral cells that in an odor-specific way align their spikes to one another. Sort of say, we are active, we're a coordinated ensemble as defined by these type synchronization of our spikes. So the question we had is where does this come from? You have this sort of respiratory theta-ish frequency coming in to the sensory system from the behavior of sniffing. Where do you get this gamma from, and how does it regulate the spike time? So this is what I'm told is a frightening depiction of the olfactory bulb. Short version is here are the receptors, glomerular layers, circuitry, which I don't get to talk about today. What I consider the first layer of computation, and the second computational layer, which I'm spending all my time on today, is of course the external plexiform layer. This is where these gamma oscillations arise from. And this has been, people have thought this since Walter Freeman in the 60s first showed that these coherent gamma oscillations across the surface of the olfactory bulb were a replicable feature of olfaction. And we've sort of known that this is something to care about ever since Mark Stopfer's work in 1997 with Sita Bhagavan and Gilles Laurent, showing that if you took away that synchronization without changing the broader spike rates, that this would have behavioral consequences. But now we're in a situation of how do we actually build this? So there's a whole literature called dynamical systems literature, which is really about how networks of cells, networks of neurons, networks of agents really can create interesting dynamical behavior. So there's a lot of variability, but there's sort of two big dynamical networks that you look to when you have gamma oscillations at about this 40 to 80 hertz rate. They're called ing and ping. Ing is basically the gamma you get from a network of all inhibitory interneurons. If they're intrinsically active and inhibit one another, and you connect them together densely enough, you'll get the emergence of gamma oscillations, that frequency being dependent on the decay time constant of the gamma A receptor. Ping is where you get excitatory neurons in forming reciprocal synapses with inhibitory interneurons. So it seems like the obvious choice, but it had problems. For the most important of these problems was that the frequency of ping also depended on the gamma A receptor time constant, but it also depends on the principal neuron's level of activation, which is great as long as all these mitral cells have about the same degree of activation, but that's just not true in olfaction, right? The different levels of activation of the mitral cells are the first principal component of what it means to be an odor. Diversity is critical. And this would sort of tear this dynamical system apart. So we needed something to stabilize this if we were going to really understand how this system generated and maintained oscillations, especially when there's zero phase oscillations spread over a physically large distance like the surface of the olfactory bulb. So Jesse again, and also Shane Peace, did some experiments also in this slice optogenetically stimulated slice mounted on multi-electric arrays. And I was going to cut to the short version of this, which is that, poor Shane, man, we didn't know this at the time. So we got these, we induced gamma oscillations, and then he tried to block them by putting on bicupene, standard gamma A blocker, didn't work. Tried gabazine, didn't work. Tried picrotoxin, didn't work. I'm ashamed to say we started making fun of him, but he was right in the end. And when we looked at it more closely, what happened was that there was a change. There was a reduction in frequency of these oscillations, but more importantly, across the field of the array, they were no longer coherent with one another. So he was blocking GABA A just fine, but the oscillations per se persisted in the individual columns, but were no longer coupled. So no longer capable of synchronizing spikes over a broad distance. And so our first thought is, how are we going to show that we successfully block the GABA A receptors? Not hard, but here we are. And so what Shane did was to, actually, sorry, Jesse did this because we did the experiments, was to put on mucimol, which is GABA A agonist. So basically turning on all the GABA A receptors, flooding everything with shunting inhibition, so the whole system shut down. And then added BMI back in to block those receptors that were being stimulated. And we got our oscillations back. So it is genuinely true that these individual columns can oscillate by themselves, even if disconnected from other columns. When I say column, I basically mean glomeruli and their associated neurons in interneurons and principal neurons. That's just showing that we get averaging across all our data, we get that oscillation back. So where does this come from? It comes essentially from the intrinsic resonance of nitric cells. I believe that they are synchronized with each other through gap junction connections, but we've not yet actually tested that. It's sort of a gap in what we've managed to do so far. But this is work by David Divison from actually 20 odd years ago, just recording from mitral cells. You can see they have these little subthreshold oscillations. But there's greater implications to this. These are resonant neurons. In addition to having these little subthreshold oscillations, have sharply preferable spike frequencies. They don't just increase spike frequencies, you go up. They have a kind of an attractor for certain frequencies that they like to fire at. And so you'll see that greater intensity of firing tends to be larger bursts rather than increased spike rates. Dan Rubin and I built a biophysical computational model to replicate this in 2006. There's more to it than this. There's a whole balance of currents that are of interest, but this is just to show you that you do get the same underlying behavior using the many constraints and good documentation of Davison's paper. So here's a map of what the currents look like during these oscillations. Again, this is from the model. This is the persistent sodium current, which will play a role in a moment. And the IA, which is the inactive potassium current, the calcium-dependent potassium current. Also, those are the three that really interplay to create these subthreshold oscillations in mitral cells. So they have a behavior like this naturally, and they also critically, if they receive shunting inhibition, this resets their phase. This becomes critical because now we're going to talk about a situation where these cells are oscillating by themselves, maybe these clusters oscillating by themselves. When you do have inhibitory inputs from other glomeruli through the granules, or in this case, we'll mostly talk about the granule cells, that inhibitory input will reset the intrinsic phase of these oscillations. In other words, it'll facilitate the network-dependent reset of phase and hence facilitate the synchronization of spikes between two neurons. The downside of resonant neurons is that they have their own opinions of what phase they're in. But if you have a common reset, that problem goes away. So this is a paper that's entirely detailed. So I'm going to skip to the end. This is Goshi Lee's work, another sort of biophysical model, taking that into a full network. The gist of it is that if we remove subthreshold oscillations by turning the persistent sodium current into an ohmic current. So again, the currents are the same, it's less dynamic. we sort of break down the long range and temporal coordination of these oscillations. In other words, short version is that it is this intrinsic resonance that enables a ping network to be resistant, or a ping-like network to resist being destroyed by these vastly different, these largely different strengths of vector cell input. And it coordinates successfully over a wide physical distance, despite the fact that there's relatively understructured coupling at those distances. So, so a lot of the problems inherent in the nature of our olfactory bulb circuit go away because of this resonant neurons. So we ended up calling this PRING, which basically is ping, including resonance, a pyramidal resonance interneuron network gathering. So my point up till now has just been to sort of drive home the point that this is real. These oscillations are real. They are evidence of spike timing interactions, which we and some others now have shown to be real and above what would be expected by chance. So to present this idea that the information going to the next level, which we do know is sensitive to synchronized spikes coming in, is really selective for inputs that are synchronized on this time scale. There's even a few ancillary advantages to it, like getting rid of that last bit of concentration-dependent error. So I've asserted this view, and I'm not going to talk about a model that the first version of which we published on this Intel Luihi chip. Without going into a lot of details, it's one of the sort of current crop of called neuromorphic processors. They're computer chips, like analogous to CPUs, but they are organized to work like brains do. People keep saying that. This is the next iteration forward of that principle, because it really is about inputs that are parallel, computations that are local. There is no shared memory. There is no memory bus. There is no global solution. It's entirely based upon what systems can do based solely on local and cascade information among nodes. So it's graph theory on a chip, with the added bonus that each individual node can do arbitrarily complex things. So it's much more interesting in that way than a deep network that are really just a bunch of integrated firing lines that do one very simple thing multiplied by 1,000 times. These can be arbitrarily complex. So it's a much better model for instantiating a biological system into hardware. That said, most of our current development of this model is done in regular computers. Okay, so here's the nature of the problem. And this, for me, this is foundational, and we should all be thinking about it more. So here's a fox has just killed a rabbit. It's being rewarded for this. Its face is full of the smell of rabbit. Everything's great. Some time passes. The fox is hungry again. Wants to seek out another rabbit. Typical sort of predator search effort, except there's lots of smells in the environment, and most of them aren't rabbit. Right? The idea and the problem spoken here is that there's, for every odor we have, whether it's a monomolecular odorant or a particular combination of odorants, it gets its properties through its interaction with our receptors. The similarity of odorants are because of, to the degree they occupy or activate similar groups of receptors in our nose and subsequent processing thereof. If you have other odors that compete for those same receptors, they might be antagonists. So if this is rabbit odor here, and then in the background here you have your blue receptor is blocked because pine tree, some component of pine tree is an antagonist of that receptor, so that goes away. And there's some sort of competing weak agonist or partial agonist with the pink receptors, that is diminished. But there's another sense, something from ground perhaps, that's enhancing that the receptor, the orange receptor's net activation, so now includes both fox and this other thing. And so here's your point, the sort of three-dimensional receptor space. Now this is a cartoon where there's only 3 receptors, but the point is that background odor mixing in background is a different odor as far as the activation of the primary receptor seed is. And so how do you get, if that is your point, and that's really the only information you have, how do you identify that that point is present and the rest is noise? One thing that's definitely true, as far as I'm concerned, is that you can frame that as an unsolvable problem, but solving this problem is something you can do if you have representational learning within the bowl. So I'll just take a quick segue. I suspect I don't need to convince this audience that the bulb is plastic. In case I do, the bulb is plastic. And this is work by Michelle Tong, who left the lab a few years ago. And the gist of this is doing a sort of a bog standard memory consolidation experiment that you would often do in hippocampus or isocortex in olfactory bulb. And the gist of it here is that it's an associative discrimination task where animals have to discriminate between two odors to get the rewards. And this is during their 20 trials of training, they perform better and better. And that's all great. And the two variables of interest are that half the animals were tested again 2 hours later, short-term memory, classic short-term memory, non-protein synthesis dependent recall. And half were tested 2 days later, which again classically requires protein synthesis in order for that memory not to be lost. Then the other aspect of it, 2 by 2, some were infused with vehicle into olfactory bulbs, and others were infused with K252A, which is the TRAC-B receptor blocker, BDNF receptor blocker. One of the things, sort of the classic requirement in many systems for memory consolidation. This, and then so the drug was only delivered here, and it has no effect 2 hours later, it has no effect in its vehicle, but if it is long-term, talking 2 days later, and K252A, that memory is largely forgotten. It's not quite down to chance, but it's very significantly reduced. So just another area, the bulb is plastic. It follows traditional pharmacology insofar as a single experiment can allow, and consolidation implies that these are durable memories that live in the bulb. So we're not just tweaking a neuromodulatory filter or something like that. So again, everything I'll talk about here is the EPO. So this is now, it's going to use a bit of this machine learning speak, but I do also consider this to be a model for how we should think about olfactory bulb computations. But I'll show you the raw materials of what we're doing here is that the odors we're using are actually from a wind tunnel with 72 sensors. So we only have these 72 inputs instead of the 400 odd that we enjoy. There are 10 different odorants in play. There's 2 sources of noise. There's the odor plume itself, which is sort of a quasi-Gaussian type of variance in intensity. And we experimentally add impulse noise. And this impulse noise it basically means we get our input, we replace some fraction of that input at random with random numbers. And this is meant to be the effects of unpredictable competitive odorants that are blocking or adding to, enhancing or inhibiting receptors, right? So it's this unpredictable source of error. We didn't do a lot of work on maximizing performance. This is proof of concept. Again, this is only external plexiform layer at this point. So we have our cartoon mitral cells, cartoon granule cells, sparse initial connections. So not all possible connections are made. Something that will be important here is that our metric, we are interested, there's a gamma oscillation going on, which you'll see how that becomes important. We care about spike phase with respect to it. Stronger excitation, actually I will talk about this now. So instead of there being an endogenous biophysical mechanism here, we impose this gamma inhibition on the circuit, which means that when you have a certain amount of analog activation, as that inhibition gets released, the stronger you're activated, the earlier you spike, all else being equal. Right, so basically converted an analog value into a single spike if it can be interpreted with reference to that phase window. So stronger excitation is the phase lead. And if you get feedback inhibition, or when you get feedback inhibition from granule cells, that doesn't prevent a spike or sink a spike, it delays a spike. We've done some previous work on this just to show the biophysical appropriateness of this, given the architecture of that system. These inhibitory inputs coming in on the secondary dendrites are they're pretty crappily positioned to actually sink a spike, but it's really good at just sinking enough charge to delay the spike on the time scale of a few milliseconds. So we're now creating this whole, reproducing this patterns of activity levels that we're used to into patterns of spike phase, where more activity, earlier phase, less activity, later phase, no activity, no spike. So spike phase represents analog values, but this gamma discretization, so one sniff with many gamma cycles in it, enables a sort of iterative processing, which is both useful during learning and useful during recall. Another thing that's important here, because this is something else that I want to talk about with some of you, is that, again, for biophysical reasons, mitral cells effects are independent of space. They can hit any, they don't hit all granule cells, but they can hit any granule cell in the circuit with equal probability. Granule cells inhibit mitral cells only locally. And that's biophysically correct. It's more literally correct in this model than it is in real life. But granule cells, essentially, GCs only affect their local mitral cell, but are affected by mitral cells drawn from across the ball. So if you get, these are just two odorants in that data set affecting our model here, and you see that these are 72 mitral cells, one attached to each sensor. Early phases are the ones that are strongly active, late phases are the ones that are more weakly active, and of course some are not active at all. And if there's nothing interesting going on, no plasticity, no prior learning, then this is stationary. This will, as the inhibition is released in every cycle, the same pattern of spikes will recall because the same analog levels of receptor activation are present across the system. A different odorant, toluene in this case, has a markedly different pattern. So far, so good. Spike timing-based representation, except still suffering from my criticism earlier that there's been no challenge to this yet. Of course, there's a common representation. It's a physical system, same input, same output. But what the system does, this is the most interesting, I think, from a biological point of view, part of the hypothesis, is that granule cells are really the source of configural learning in the ball. So what we have here is the idea we have this matrix of mitral cells going out to excite granule cells. And the hypothesis here is that there is regular asymmetric spike timing dependent plasticity learning rule at that synapse from mitral onto granule. And that means a few things. So here we have some, for every granule cell, there's going to be some larger than 5 number of mitral cells at start impinging onto it with relatively weak inputs. And you'll get some input here like this acetone input here. So some number of these mitral cells are going to spike. And so we have a copy here of these 10 mitral cells are spiking, and the spike timing dependent plasticity rule is drawn originally from the hippocampus. It also has been biologically shown in some parts of isocortex. It's not been shown in bull, but it's so that's it's hypothetical. But again, it's this and similar rules are fairly common in the brain. So we have these 10 inputs, 10 mitral inputs to a granule cell, and eventually they accumulate enough activity to evoke a spike in that granule cell. And by the spike timing dependent plasticity rule, those presynaptic neurons that were causal have their strengths increased. Those that are non-causal, those that happen but too late, they follow the postsynaptic spike, they're weakened. And so that's standard, and you could strengthen these and weaken these, except there's another moving part. And that's that because these are stronger, now they're more effective in contributing to the depolarization and the firing of the granule cell. So now it takes fewer of them to make this spike. So over time, you get a precession of the granule cell spike, where now in this example here, it just takes 2 of these two contingent mitral cells to spike for that granular cell, and the others are all weakened. So some of these in the middle were first strengthened and then weakened when they fell off the trail. And you end up with this, where you've gone from a situation where there was a large number of weak inputs to a mitral cell to a small number of strong inputs. And that's considered a differentiating event that stays true. That granular cell is now sensitive forevermore to that particular combination of mitral cell inputs. It has a more specific receptive field than any of the individual mitral cells. And you only build such configural receptor fields for things that actually exist, right? So this learning irreversibly differentiates these GCs, and this is where the need for adult neurogenesis comes in, because of course, over time you begin to deplete this pool of GCs. You need to restore. So if you're going to use this mechanism to learn arbitrary new things about the world, you have to replace your raw materials, which is exactly what sort of adult stem cell neurogenesis is good for. If you're mathematically kind, you might have seen that if there wasn't a sort of other piece to this, you proceed to winner take all, and granular cells wouldn't be interesting. The way we can cap this to not go to winner take all, but to K winners take all, where there's some 2 here, but more likely 10, 15, 70, 150 inputs. That can be controlled by the threshold of the granular cell and by caps put on the weight of any one synapse. So that's at least in the model, is a fairly easy variable to manage. And in fact, in the more sophisticated models that we're doing now, based on what I'm showing you today, heterogeneity of granular cells is important. You want to have some that are relatively low order and some that are relatively high order. Some that sort of say, okay, I think X might be here, and some that are like, I only fire when I know it's X. So the broader version of this figure is here. You start out with a weak, a sparse but uniform field to a field where there are very specific, strong inputs that represent that granular cell's new receptor field. This is maybe less interesting from an olfactory point of view because it's really about the Luigi chip. But of course, we need an inhibitory learning rule too. The broader sense is that once these granular cells have learned these higher order configurable receptor fields, they deploy their inhibition appropriately onto the mitral cell, so as to shape their spike timing to pull representations closer to them, closer to what they're expecting. This is one way in which that happens. It happens to be, they've been very efficient on the first belief. I don't think this is as biologically plausible directly as the excitatory. It's essentially that at the inhibitory synapse, based on the inputs it gets, it learns when it should fire, or learns when the cell that it is inhibiting fires, and adjusts its level of inhibition to match that. So during learning, it's saying, I'm going to make what's already true, true, so that when there's noise, I can push towards that state. It essentially forms a fixed point attractor in that language. And so what you see then is, here's again the stationary toluene signal up here. And if we add included toluene, so now we've replaced 60% of the inputs with random numbers. So it's a jumble. It looks like something very, very different. But we then give this included toluene to the network, and over a few gamma cycles, so still within one sniff, still within one presentation, it evolves back towards the toluene representation. This is just another example of that in a naive network. The noisy signal, which is in the blue dots, just stays the noisy signal. In a trained network, the noisy signal is drawn towards the learned signal. The secret sauce underlying which way it goes is really a property of high dimensional space. When you have a large number of receptors, like R400 or vices 1,000, even the 72 here, you can be, if you have a representation that's sort of clean, you can be really different from that without becoming close to anything else. space. In high-dimensional spaces, most things are really, really far apart from each other. So that's, that's kind of the secret sauce underlying why this works. That's the magic of why I'm converging onto this representation instead of the other 9 that network has been simultaneously trained on. So I want to point out from a machine learning standpoint, this, I guess I'll say the next slide. Okay, I guess we will say that. This, what's very normal for an animal, but very, very difficult for a machine, is that this network based on olfactory bulb can learn, it learns very quickly. You can learn a new odor in 1-shot learning. You can get a little bit better if you do a few shot learning. That means like 2, 3, 5, not 18,000 like a good deep network will require. It can also learn things in sequence. Like you learn, you see the choline representation, sorry, the aldehyde representation, ammonia, butanol in sequence, and the new learning does not disrupt the old learning. Again, easy for animals, very difficult for machines. Thank you. And so, but, and this is a map of the performance with respect to that, what proportion of the inputs get included. It starts dropping off when you get above about 60% inclusion. But I bring this up specifically to point out that there's, this is just like a taste of the more interesting factors of the biological system. Even if you have 90% inclusion, which you can't really, you won't really do get any fix with this network. But if the if you can alter the excitability of some fraction of the correct granule cells, perhaps via their puriform feedback, some contextual input, like I'm in a bakery, it's more likely that I'll be smelling bread. Then, depending on how much of the granule cells you can prime that way, you can get your performance back. The point is you can have an alarmingly small amount of information if you have ancillary, sort of non-chemosensory information to help prime it. I will skip this. It's basically just saying that we add more and more and more noise and we fall off gracefully. And this is just me saying, this is basically quantifying several of the things I've said. In the performance of this network, this olfactory bulb inspired network performs considerably better than a deep autoencoder. Given that the deep autoencoder has also been trained on a limited instruction set. So this is one trial learning for the bulb, literally one presentation for our network. The deep autoencoder gets thousands of presentations, but only of the one thing. So in other words, it doesn't get to peak at the correct future variance. And if it does get to peak at the correct future variance, which is the proper way to train a deep network, then eventually it'll equal and surpass this after thousands of training trials. But the niche of this, just like with animals, is to learn things quickly and efficiently and be able to learn over time without having your older representations destroyed like happens in standard deep networks. This is all sort of points that are more relevant to the machine learning community where you're trying to say, this is another way to do machine learning. These biomimetic systems are advantageous for these particular approaches. That's essentially the end. I've got points I want to talk about here. There's an interesting backstory, which is I'm happy to talk about because it's quite presentation material yet. It's more like sit down and talk about. I can show a couple things about how a richer version of this one-shot learning is category learning. The actual, the idea that in the real biological system and in our more sophisticated current models, this network in the olfactory bulb is really about carving out categories that correctly represent the degree of real-world variance that constitutes a particular odor stimulus. And following that, sort of admits the formation of within category hierarchies, so that you can, with position in that category being amenable to the speed accuracy trade-off, or speed precision trade-off, really, where you can get a really fast response. This is an orange, and given the signal to noise your inputs and enough time, you can say, that is a slightly overripe relation, for example. So in other words, ways to tweak this system to experience more of the kind of richness of a fully cognitive biological sensory system. Everything that we've we've come to enjoy as possessors of such systems. And I think nothing here have I not said already, so why don't I close it with that? Thank my laboratory and funders, and if this is like a one-stop shop for how we think about things, if anybody's interested in that frontier statement. Thank you for being here.
I'll ask a question. And it's probably because I missed it. But it wasn't clear to me as you were discussing the importance of the synchrony of the spiking, how that allows for adaptation. It seems like, in my naive thinking, adaptation wouldn't occur then because that primary spiking is going to always be there.
Right, okay, good. So this is actually a good planting question. So what are the weaknesses of the current operation, right? Actually, both in that version one I just showed, and even still in what we're working on right now, is that it picks one thing out of the mix. It picks one thing, and everything else is by definition noise. Real sensory systems, you can sniff and pick out more than one thing, that are simultaneously present. And again, I should point out that what the category model does is emphasize that individual noses are very different by virtue of your experience. So how much you can pick out, how much you can segregate depends on an individual's experience and training. You develop your own nose through these sort of plasticity mechanisms. But adaptation, in both in literal receptor adaptation and in that sort of first synapse to drop, dopamine D2 receptor feedback. I don't know if we literally call that adaptation, depending, but same sort of thing, desensitization. Quality specific desensitization is where I think where we are going to go to solve that problem. In other words, once you've identified X, you take advantage of the adaptation to say, once X is gone, what's left? And by the same sort of high dimensional logic, enough should be left to give you a couple more things, depending, of course, on how overlapping they are, what concentrations are. That's where I'm not sure if I literally think that adaptation of receptor neurons, I would think would be independent of the sort of configural recognition. But maybe not. Maybe once you get up to the bulb, of course, all bets are off because there's a lot of projections back from deep bulb and from piriform cortex that could, in principle, help you target sort of configural odor specific patterns of adaptation.
I think in two parts. This part is what I do remember the speaker talking about balancing the fucking bug actually or restoration bug. And I don't know if you read it or not. It was from the lab in Iceberg.
So I'm sorry, you're talking about like hippocampus jovi. Sorry.
From the hippocampus, I think they actually show something like gamma oscillation driven by hippocampal, if the hippocampus being synchronized with respiration and having an effect on the functional as well. My second question, I think it's related to that, is like how gamma oscillation with the bulb actually interact with synchronized with general gamma oscillation.
Okay.
That there are internal triggers, how they can.
Right, yeah. So, yeah, gamma oscillations local to bulb. And, their output is essentially this control over spike phase. The next phase, of course, is that you sort of fall into this beta, which, again, one of the things that are stack is to try and do the biophysical models to sort of see if this plays out, but it seems fairly straightforward that once you get piriform cortex in a certain state, which of course feeds back on granule cells, then you go from mitral granule, mitral granule, mitral granule to mitral granule and pyramidal and piriform pyramidal, which will take a little longer. That comes back down here, extends the period of time that granule cell inhibition is being delivered, which before freeing the mitral cells again, which is sort of you just slow down because you've got a third element, which is a cascade of a couple cells at least in the mix. And so, in that case, we know that the beta band oscillations are meaningfully coherent. The gamma R2 is I suspect it's just the bulb's going gamma, 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 and the puriform quartz is like, whoa, 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 until it starts, participating, and then it's a genuine dynamical system of beta. This isn't always true, and gamma and beta aren't categorically different, right? They're like historically dependent frequency bands. We can't think too seriously, but so this does leak over into things called gamma oscillations, but much of the information about, or the work on inter-areal coordination, communication through coherence is in beta band. It's sort of associated with like attentional networks. It's like, I'm listening to you right now, I'm listening to you right now, through virtue of, area X is synchronizing with area Y versus area Z, as opposed to all three synchronizing at once as a way of targeting information, despite the fact that the physical connections, the weights presumably remain roughly constant. So this is a little bit dancing around. So in terms of, there are some parts of the visual system that I do not remember where the inter-areal coordination is called gamma, and I don't really know what the frequencies are. But I suspect it would not be importantly different. I mean, details could differ, of course. As far as the hippocampal projection, do I have this right? Hippocampal projection to bulb?
I don't think it,
That is a thing, but it's not.
I just remember, the people mentioned that there is some, I don't know if it's in the Peterson for this, there's a level of hippocampal, if I recall.
Oh, yes, there's lots of coordination. I mean, so, bulb, piriform, entorhinal, hippocampus is the big one, although it's long. A little bit of bulb, entorhinal, that doesn't really stop off in piriform. The bulb feeds, there's a little medial, in mammals at least, a medial projection from the bulb that goes up through the inducium museum. It used to go to the hippocampus back when the hippocampus included what's now called the anterior hippocampal continuation, but basically goes into that neighborhood. It's not big. I don't know anything about what it does. Hippocampus comes down, since the ventral hippocampus projects pretty powerfully to the, I think it gets to the bulb, but mostly to the anterior olfactory nucleus, which of course talks to bulb a lot. So there's loops there. Now Leslie Kay talked about theta band coherence, so roughly respiratory frequency coherence between the two. I don't recall exactly what the.
But then connected to that, it would be interesting to see if this loop might be involved in what we call categorical.
Yeah.
Because you said that it's, experience driven, and it's driven, like, I don't want to talk about people.
Yeah, so actually we are, there's so many ways to go with this. So we're actually very excited right now about this ventral hippocampal to AON projection. That's sort of, the upshot of that is, first of all, sort of the most exciting way to talk about that is to say that even at the very first synapse, the signal that the bulb is meeting is not an odor signal. It's an odor in context. There's the olfactory information coming in from the epithelium, obviously, but it's strongly affected by these sort of visual context and social context that are coming down into AON from the ventral hippocampus. And so what mitral cells do is not independent of these things. And so that's obviously in the real world of actual animals with all their rich complexity, that's clearly going to be part of what we're considering category formation, right? Also, we do know, unlike this model, but like our upcoming ones, you don't really, piriform cortical feedback on granular cells is important for their plasticity and differentiation. And so, so that's another thing. It's not just this sort of physical quality specific categorization. It's very much in tune with what you would end up calling a category by virtue of your broad experience. Things that mean the same thing. Right now, it's a little hand wavy, but in a few years, I'd like to be able to be very concrete about
So we've got time for like one more question. There was a question from Kevin Bolding in the chat. 1 second. Sorry for the echo.
Let's get one last question.
Yep. So Kevin Bolden's question was, much of the discussion about a pairwise synchrony code seemed to refer to gamma, but the mitral cells were resonant at beta. If one wants to look for evidence of synchrony code in vivo, is that the
Vivo,
correct pronunciation? Okay. Would you look at the spikes beta phase or gamma phase?
Yeah, my opinion is that that if it's during gamma, it'll work in gamma. And once you put the, once peripheral cortex inserts itself